I had to give a talk to the Mathematical Biology Society. I gave one in Canada about a month ago, and the local one asked me to do it again. So forget the second part of the title. With only 15 minutes, I'm only going to show you the results that we get, to try to model the growing and dividing of the minimal cell. All I will tell you is that it takes a lot of experiments, theory, and a lot of new computational approaches to do this. And that list of participants in this product, or almost all of them are co-authors on this paper that's under review at the moment, and Cell. And so let me then go on and tell you, this is our minimal cell. As you all know, it's very small. John gave you the statistics on the breakdown of the gene map here. Those unclear ones, very important. As John pointed out, there's at least 60 of them, or 50, I would say, is when the number I came up with are essential. But the good thing is, with the recent Nobel Prize in AlphaFold, you certainly can predict structures of all of them. And all those papers down below, the majority of them are with JCVI, and we just looked at each of the subsystems one after another till we put it all together in Cell 2022, but it didn't grow, and the DNA didn't move, and the ribosome stayed in the same place that we got from the tomograms, both from Elizabeth Vila, and now it turns out the Chan Zuckerberg people have produced over 100 cryo-electron tomograms of this system. So we're getting so much data, we felt it was ripe to try and model the cell. And our goal, as I said, was try to evolve it over the full cell cycle of almost 2 hours, and to try to gain insight into some of those minimal rules of life that John talked about. To do this, it's for a computational person, it's quite straightforward. We need an initial state. That means we got to know who's essential, and everything we've included in this model. How about the numbers of proteins of each type, the lipid types, the small molecules, the metabolites, and an estimate of the mRNA initially, and any type of kinetic data you can give. We are also lucky for one of those previous papers with Ramos Dame. We had a DNA contact map, so we knew that it had very little supercoiling and domain structure. It turns out that made it easier for us to move it through the cell. And then you have to have known all the interactions and the diffusion coefficients. And we were very lucky. I think decades of these dedicated biochemists have really explained almost every reaction that we needed in the cell. And then we have to evolve the state of the cell from time t equal to 0 through that 110 minutes. And we do it using what the mathematicians now love. It's a whole new area of mathematical biology, these hybrid stochastic deterministic methods. All I will tell you is that we impose a lattice on our cell, and that allows us to dictate certain rules in each of those sublattices. So things can diffuse between a subvolume, or they can react in a subvolume, or you can use it for things that say, oh, okay, this cannot go out of the cell, or this can come into the cell. And we're thankful to our partners at NVIDIA, because we use 2 of their very best GPUs that they developed for machine learning. And so we're able to reproduce and grow the cell and divide it in 4 days. And then you can get any time-dependent quantity you want, and compare as many of those you can to experiments. And the rest of them we just have to view as predictions. So I won't bother you with the algorithm, but this is just to show you again that we impose a lattice, and you do have different excluded volume conditions. So for example, if you put a ribosome somewhere that's normally more than 1 lattice site, it has special rules how this iceberg can move through the cell. Same way with the DNA. And we explain this through various journals, one for the physicists, one for the computer scientists in the past. But whenever you mix methods, you have to decide on when do you communicate between them and what do you communicate. And that was in itself its own publication. And now I think you all know you can look at the metabolism of the cell on 1 page, even though we had a very gifted postdoc, Marion Breuer, who's a professor in Europe now. He was the lead author of that ELIFE paper. That's where we introduced SYN3A. And we did that because as beautiful as SYN30 was, it did have irregular morphology upon cell division, and no biophysicist wants to try to image something that keeps changing shape. So the changes that they made in eLife, which is essentially put back in about 19 genes, made it a more consistent morphology, but you still get some interesting exceptions. And then each of those students in my group tackled coming up with the kinetic parameters. And you do have to use machine learning, but you have to do at the end a free energy analysis. And if you look at the Cell 2022 paper, that the SI is just full of how we did that. All right, we're really thankful to Hamilton Smith. I think he had tried to get a grant to explain how DNA replication started. It was rejected, but we believed Ham, and we looked also for those 3 high affinity sites that are over here near the origin, and you'll see 3 of them are right together. That's where the that's where the DNA A binds to single strand. We think it twists it open, and then you start having another domain of DNA A bind to it and form this long filament, so you can get the machinery for the replosome machinery in there, and replicates it in both directions. So we even checked this with a more recent Nobel Prize. We put in a segment of the DNA with and without those 3 signatures, put in 3 copies of the DNA A, and boy, it did find the right binding site. And it even gives you, in addition to that, some insight into how the DNA A molecules interact with one another. Then how do we, once we get DNA initiated, the replication, we have a simple model called beads on a string that is further advanced by one of my students, and then Andrew Mayton, a new physics student in my group. And we were able then to grow it, but you need definitely both of those proteins that are essential proteins that they could not remove when they reduced it, the SYN3A, from the mycoplasma mycoides, and that is the SMC, it acts as a dimer, and topoisomerases, because they have to help break up the entanglement. And for the kinetics of how much DNA is ratcheted, we use a lot of the beautiful studies from Kees Dekker in the Netherlands. And then we still do not have a good model. It's almost there for the formation of the septum, but we had the brilliant work by Tae-Kip Ha, where he labeled the DNA, the membrane, and then the FTSZ, which is in the septum, and he could identify when it, at the beginning, when it's spherical, when it gets into its prolate state, and when it gets into the dividing state. And it looked fairly symmetric to us, so we imposed that. But I also put in the corner here some interesting new structures that he saw, and they also saw it in some of the tomogram, these cells within cells. So that brings up a whole theory of how the surface to volume ratio has to be during the growth period. So, but we did assume a symmetric cell division. It doesn't come out of our model. We put it into the model, but the rate at which it separates symmetrically is determined by the kinetic parameters that we had developed in our lipid metabolism. And thanks to the brilliant work of James Sense at Dresden and Kim Wise at JCVI, we actually know the composition of it in the medium that it was grown in. And so we were able to hit that spot on. In fact, James came here to the U of I, and we had a summer school where the students repeated it and also got the doubling time that we'd seen before. And I hope he's speaking at this meeting, because he's done some brilliant new experiments where you don't even have to feed it the fatty acids. You feed it particular lipids. It makes it for a little bit more unstable shell, but it's beautiful work. And then now here are the beginning of our results. We grow it. You see the bottom of the axis here on the left is time. And then we look at how long it takes to double the DNA, how long it takes to double the volume, and then how long it takes to double the surface area. So that sort of gives you the steps in the growth here. And if you look at those periods, you can calculate the so-called B, C, and D periods, and we get the ratio of origin to term to be 1.28. We checked that with yet another experiment, but this is independent, the DNA sequencing experiment, and there we get 1.2 is that ratio. So I really should have rounded ours to 1.3, but I wanted to say we feel we get really close. And again, this is really due to some beautiful work by a colleague here at the U of I, Angad Mehta, and his students, who can grow the cell for us. In fact, they grow it so well that allows Take Kipan, his postdoc, to come in and go to our shared microscopy labs and then visualize them. So here's the movie of the cell growing and dividing. On the one on the left, I'm just showing you the ribosomes, those little bouncing balls, and I think you can see the other DNA beginning to come in. It's expanding at a certain rate, again, determined by our lipid metabolism. So you have to know how many lipids you have and how many membrane proteins you have and assign an area to them. And then on the right, we've sort of, projected everything onto those lattice sites, including the DNA. Now the ribosomes are going to change color depending on whether they're processing a messenger or not. And then the membrane proteins bringing in the sugars, the PTSG, are shown at the top. So let me go ahead and start this. And you see it's the same process. It's just we have to decide what we show because there is so much going on here. To try and capture it all in one movie is almost impossible. But I think you can see already the other DNA coming in, and it starts dividing. The time frames that we used here for the movie on the right a little bit different than the one on the left. But now you can check, is the cell division, how symmetric is it? So let's go look at the panel on the right, and let's look at the ribosomes, the degradasomes, the PTSG, and a protein in a metabolic pathway. And it all looks about not quite 50-50, a little off, but we didn't expect it to be otherwise. And then you can zoom in on a particular one. So I think what I like is the ribosome count, because actually at the end of the cell cycle, not every ribosome is really complete. And through cryo-electron tomography, they're beginning to see how many sort of separate subunits are available. So A and D sort of are things that we predict, and then the D can be checked. And then if you look at F, that has the replication initiation, you'll see that it's pretty much bunched around under 10 minutes, maybe 5 minutes. But there was one in the replicates that we looked at that didn't start to replicate the DNA until 45 minutes, but it was able to complete it. But its cycle was much longer than the cycle that we typically measure, that compares very well to the experiments. You can also get I, the mRNA lifetimes, and then we all look at how many proteins we reproduce, do we double it? And then the last two are really great because it brings home the stochastic nature of genetic information processing. So we're looking at the messengers that are being formed for a particular gene. It's the one that codes for DNA A, and then you can also see the proteins. And then we want to point out that this cell has trouble maintaining its pool sizes of all the building blocks for the macromolecules, and there can be a dip in the transcription rate. And finally, just to show you where all the time goes to do these things, on the chart on the right, for us to do this thing in 4 to 5 days, and for 1 second, it takes 18 seconds just to do that reaction diffusion part, to move the DNA, to move the ribosomes, which are like icebergs moving through there, and then to take care of the metabolism, and then write out the things that are frequent enough that we can make the movies that we show you. And then finally, one check that we as theoreticians use, we try and take our model on the left, which has 200,000 particles, and convert it into an all-atom simulation. And this is, again, where you really start using the AlphaFold 3 and structures and EMN and cryo-EM. And when you do it, you have about 2 billion, but MD always has femtosecond time steps. So you can only sort of examine a local state that you want to transfer over. But that is done nicely with our collaborators, Marek in the Netherlands and Eric Lindau in Stockholm. And then finally, when we had our first workshop for the computational people, we even made a mini cell, and they tried to simulate it for about 100 nanoseconds, which is about all they could do. And then some of the trajectories we knew still had the cells dying or having it, the doubling time get excessively long. And then we turned to the computer scientists to help us analyze the phenotypes in those populations. And then, as I said, James comes in and tells us, oh, the membrane, it has this in it, cholesterol, PC, PG, sphingomyelin, cardiolipids, and they can measure the amount. And we did that twice. Most recently, we had a summer school in July. So you're seeing pictures from the various activities we do here at our NSF Science and Technology Center for Quantitative Cell Biology. And if you're interested, one of the things we have to do as an NSF center is have an outreach that has impact. So every cell that we make, whether it's the minimal cell or we get them from imaging, like say from the yeast cells or from human cancer cells, we put into Minecraft. And I'll just show you quickly, since John is here, the one where we dive into the all atom version of the minimal cell, and you're seeing the control pad. And the student was supposed to just break away the membrane and show you the DNA and the cytoplasmic proteins. You're seeing some of the membrane proteins, but instead he put a roller coaster into it to ride along the DNA. And I'm telling you, when you give talks, you better make this one of your last slides, because this is what everybody wants to see again and again and again. And then you're seeing very nicely the DNA through it being twisting and turning, and the double helices there. So let me just finish by just showing you who is all involved in our STC. And you see John's picture there on the left with our other fantastic, we call them our industry partners, NVIDIA, and then also an imaging institute, because we need to look at subcellular dynamics. And some of our other partner institutes are the Chan Zuckerberg Biohub, and they're really helpful, along with the head of our supercomputer center, Bill Gropp. And finally, this is the mission of our STC, which is to bring these cells to life on a computer and or in Minecraft. But we bring to it the awesome new experimental techniques, besides developing these 4D models. And it's MinFlux to look at the subcellular dynamics. We're trying to develop a technique called label-free in-cell IR. It's AFM IR microscopy with machine learning to tell us about the spatial mapping of the metabolites through the cell. It's going to work best when we move on to eukaryotic cells. And then, of course, to get at the cell shape and architecture, we really rely a lot on the data coming in from cryo-electron tomography, as well as soft X-ray tomography. Those give you really insight on the whole cell, but we use everything from the molecular level into the cellular level. All right, and again, I just want to show you all the people that are in the center. There's the faculty as well as their students who have helped us with the minimal cell, and now we're moving on to our first sort of what I call the minimum eukaryotic system, which is yeast. And we're very, very fortunate that it all got started with the JCVI. We learned so much, and so much basic cell biology for that. It really guides us how we go after even something like yeast by just looking first for its essential genes, which is something I'm very proud to say was in that first 2019 paper, eLife, that Marion Boyer put together with the colleagues from JCVI and a very good colleague in San Diego who did the proteomics for us. And then, of course, JCVI, I think, were one of the leaders in doing identifying what genes are essential, which is absolutely important if you're going to go on and try and apply any of these techniques to other systems. Thank you very much.
Sam, that's always dazzling. The show keeps getting better and better.
I hear you, but I can't see you. Let me get rid of my screen. Okay.
So I'm opening it for questions, but I did want to mention to you that and it will be explained in the talk that is given later today by our group. We now know we have a measurement of how much glucose per cell is used by cells in exponential growth. It's a little different than the estimate that we based it on from mycoplasma pneumoniae in 2019. So you'll get those data to you and other groups, but this might require a little modification of the models.
Yeah, one thing, John, we cannot get from our model, because we assume the symmetric cell growth. that would probably, that glucose would definitely have an effect on the surface area and volume ratios. And you cannot get those cells within cells, even from using the Helfrich Hamiltonian that I think James Pelletier first brought up in one of our discussions. You have you put that ratio in, and if you just use the values that we have from the basic model, you do also get symmetric growth. It just wasn't the cells as they divided looked more like teardrops. So it didn't look like any of the images we saw. But if you put in the effect of membrane proteins and you get a variation in that time course of that ratio of surface area to volume, you could also probably start investigating why do you get cells within cells. James Sense sees it when he changes the membrane composition of the lipids. He's very proud of that. And if he's speaking, this is new results that he showed us at the summer school. Really, this is fascinating science because I think lipidomics, I always say you need affirmative action for lipidomics.
The questions for Zan.
Hi, Zan. James Pelletier, and I'll help
hi,
you.
James.
Great to see you in a beautiful work as always. Thinking about your chromosome model, I'm curious in the model, can you remind me what forces coordinate chromosome segregation with cell division? I remember in addition to loop extrusion, there were other forces. And then a related question is, have you ever tried to simulate other geometries? For example, can a chromosome cross a constricted neck into a space where there is not yet a chromosome? For example, if the division were to start before, I was thinking about how the chromosomes enter into these different non-symmetrical geometries.
Mm-hmm. Now that's a good question, James. So